Who's doing them in the fall? Who's doing them in the fall? He is, oh good. I'll make sure he fills up his spring. Hey, how you doing?
All right, I think we're on TV.
Forewarned.
He's not trying to be rude. All right. I think we are ready to start, right? Okay. So our special speaker today is Dr. Ronke Olabisi. Dr. Olabisi is a postdoctoral research associate from Rice University, and also currently the holder of a very prestigious National Space Biomedical Research Institute postdoctoral fellowship. The NSBRI is the biomedical research arm of NASA. So Dr. Olabisi first graduated with a bachelor's degree from MIT in mechanical engineering. She went on to do 2 master's degrees, one in mechanical engineering and one in aerospace engineering at University of Michigan. And then she went on to University of Wisconsin-Madison to do a PhD in biomedical engineering. And she has a distinction of being the first African American to graduate with a PhD from Wisconsin. Then she went on to, after a year of a postdocing with Wisconsin, she went on to Rice, where she has been till now. Her major work is in tissue engineering and particularly in growing bone cells, and she's going to talk to us a bit about that today.
Okay. Can you hear me? Or how's that? Okay. Thank you for the introduction. I'm going to be talking to you today about my work with bone formation. Sorry. Okay. In the U.S., there are 500,000 bone grafts performed annually, and worldwide there's over 2 million. The indications for bone grafts are bone defects such as fracture. In this case, they would take a bone graft and fill the hole. When you have a defect because of disease, and in this case they would fill this with bone graft. Also, in cases of spinal fusion, for scoliosis, what doctors would do to straighten the spine is they would take metal rods and hooks to straighten it. They would then fuse the spine to prevent it from curving again, and use bone grafts to get that to happen. When you have a fracture, in the case of spinal fracture here, you want to fuse the spine to stabilize it, or when you have a herniated disc. And the reason that they would fuse this spine is the herniated disc will not repair. And here, the arrow is pointing to where it's pushing on the spine. It's causing pain and weakness there. And the goal is to take out that damaged disc that will never repair, to replace it with bone graft, and to hopefully fuse the spine like that so there will be no more pain. And this is not an actual case. This is an idealized case of what doctors are going for when they initiate a bone graft. The current grafts available clinically range from autologous, allogeneic, xenogenic, alloplastic. Autologous simply means that it's taken from yourself, and generally it's taken from the iliac crest, and this is just the hip. They place it where it needs to go, and that's autologous because it came from you. The allogeneic grafts are from cadavers, and these are freeze-dried and processed or fresh frozen and processed, and they can be taken from elsewhere because you're not limited by the amount that you can harvest. Xenogenic grafts simply means that it's from a donor from another species. Generally used today are bovine, which is cows, or pigs, or in some cases coral is really highly processed to resemble bone. Alloplastic grafts account for less than 15% of the grafts available in the United States. And here is an image of polymeric alloplastic grafts that are available. So autografts are currently the gold standard in surgery today. The reason that surgeons like them is because they outperform all the other grafts available. You don't have any rejection because it's from yourself. It forms bone and generally incorporates well into its new location in the body. However, the disadvantage is that it requires a pretty invasive additional surgery. As you can see, they cut everything open to reach this piece of your hip and pull it out to put it elsewhere. There's a limited quantity available. You can't just keep going back to parts of your bone and taking pieces out. And sometimes patients have complained of persistent pain at the donor site for up to 2 years. And for these reasons, alternatives to the autograft are highly sought. If you're going to do a biomaterial approach, the biomaterial must be at least as good as the autograft. And the 4 main features of the autograft that people like are that it's osteoconductive in that bone can migrate through the graft. It's osteoinductive in that it causes progenitor or undifferentiated stem cells to head into the differentiation role of bone cells. It's osteogenic in that it causes bone cells to make bone, and that the body can degrade it, allowing for the replacement of it by new bone. Now, tissue engineering is also another field that has approached the alternative to the autograft. And tissue engineering is a field that uses scaffolds, cells, and bioactive molecules to try to grow new tissue. Gene therapy is also a field that has been investigating autograft replacements. And their approach is by modifying the cell's genes in order to get your body to grow the new tissue. Tissue engineering strategies generally fall into in vitro or in vivo categories. And with in vitro, essentially, if you follow this path around, what they'll do is they'll take cells from a person, and then they'll expand them in tissue culture. And then they'll take these cells, once they've been expanded and you have enough, and they'll put them in a scaffold. Now, these are bioactive molecules. They could be signal peptides. They could be hormones. Sorry about that. And they put those in the scaffold as well. And then they'll take the scaffold and generally put it into a bioreactor, and the bioreactor will grow this tissue, and from that point you put it in the body. This is an in vitro scheme of creating tissue through tissue engineering modalities. When you're using an in vitro scheme, the things that you generally tend to change are the bioreactor conditions, because some cells respond to stresses. Some cells respond to oxygen tension, like chondrocytes, and that will cause them to differentiate. Or bioactive molecules. Some cells will be caused to differentiate by these molecules, and you can incorporate those into your graft to get the desired effect. With in vivo strategies, you tend to use all the ingredients that you would in the in vitro strategy, except you use the body as the incubator. So in some cases, they just take the bioactive molecules and put those directly into the body, so this straight into here, and hope that the body will do the rest. In other cases, they just will transplant cells, usually from either another part of your body or from another host, and hope that the cells, once in their new location, will do their job. In other cases, they'll just transfer the scaffold, and it would be this scaffold without cells in it. Most likely it has bioactive molecules added as well, and then they'll skip step 4 and go straight into the body, or a fully cell-seeded scaffold implantation, and so it would follow all of these but skip the growth in the bioreactor and just go straight into the body. So of the cells that are used with tissue engineering methods, they're all taken from either a human or an animal, and they range from stem cells to fibroblasts to osteoblasts. So I'm really not going to talk about those here, but I'm going to talk to you about the tissue engineering scaffolds. And so the scaffolds can generally be broken up into several groups. You have natural scaffolds, which can come from bone, like collagen, or demineralized bone matrix, or decellularized bone. Or sometimes they come from plants, like wood, Some researchers have used wood as a scaffold for bone growth. Or another group of researchers used cornstarch that they blended with an ethylene vinyl alcohol. And you've got organic polymers, polyanhydrides with polyamides. They're very biocompatible, and with the polyamide, they have the compressive and tensile strength for bone applications. They're generally used in drug delivery. They're linear aliphatic polyesters, polyglycolic acid, polylactic acid, and their copolymer, polylactic co-glycolic acid. And polyglycolic acid will degrade very fast, about 2 to 4 weeks, and polylactic acid degrades much more slowly. And when you blend them, you can alter how quickly it will degrade, and these tend to be used in sutures. There are tyrosine-derived polymers like polycarbonates, which are relatively new compared to the others, and these are shown to be pretty bioresorbable and friendly in the body, somewhat bioinert, and they are degradable, and so they're coming on the scene more. You've got your inorganic constructs, which one of the more popular ones is hydroxyapatite. And hydroxyapatite is popular because it is what the mineral portion of your bone is made of. Because hydroxyapatite has calcium and phosphorus in it, calcium phosphates are also used as a construct, and they've also taken ceramics and glasses and functionalized them with bioactive molecules so that they too can form or are used as a tissue engineering scaffold. And finally, there are hydrogels. You can have synthetic ones like polyethylene glycol or PEG, and natural ones like alginate and agarose. And PEG can be used to encapsulate cells. More difficult, you can encapsulate them in alginate, and even more difficult in agarose, but they can be molded into many different shapes. And so the other method that I talked to you where they're looking at alternatives to the autograft was gene therapy. And this schematic kind of shows the general overview of how gene therapy works. So here, first you have the viral DNA, and you insert your new gene that you want to be used. And so in gene therapy, when they're talking about a vector, what they simply mean is a way of getting your gene of interest into the cell. And because viruses have evolved over years to do this, they do it much more efficiently than any artificial method. So in gene therapy, they tend to take advantage of the virus to transfer genes into the cell, which is also called transduction. So here, you would modify your gene. You would put it in your viral vector. In this case, it's an adenoviral vector. And the vector will then bind, the virus will bind to the cell surface. The cell will then invaginate and encapsulate the vector in a vesicle. And this vesicle will break down, releasing the vector. Now the vector is free to bind to the nucleus, where it will inject the new gene, where the cell will then use that gene to make the protein of interest. So again, there are two different modalities of adenoviral gene therapy. You've got in vivo and ex vivo. And in vivo simply means that the genetic modification is occurring within the body, whereas ex vivo means that the modification is happening outside of the body. And so in vivo, what they do is they'll take your virus and they'll inject it directly into the body. The virus will deliver the genes to the host person's cells, and the modified cells will then make the protein. With ex vivo gene therapy, the adenovirus is incubated with cells, and these cells are then harvested and injected into the body. The reason that people tend to be more comfortable with ex vivo is that you can be exactly sure of which cells you're transducing, and you don't have to worry as much about systemic infection of the virus. There's less risk. So I've talked to you about how we're trying to repair bone. Now I'm going to explain to you how bone repairs itself. So let's say you have a fracture. Initially what happens is the substance that I'll explain later, called BMP2, is released from the matrix of the bone where it is stored. And it has an effect on MSCs, which are mesenchymal stem cells. It causes them to differentiate into chondrocytes, and pre-osteoblasts. And chondrocytes will lay down a cartilaginous callus that fixes the bone fragments. The pre-osteoblasts will continue to develop into osteoblasts, where they will mineralize this callus, forming new bone. Osteoblasts release a substance called receptor activator for nuclear kappa B ligand, or RANKL. And RANKL has an effect on monocytes. It causes them to merge and differentiate into osteoclasts. Now, osteoclasts will release cathepsin K, which is a protease, and hydrochloric acid. Now, it'll use these to degrade both the organic collagen portion of the bone and the mineral portion of the bone. And so osteoclasts can use this to remodel the bone into the appropriate shape. So I said I'd talk to you about BMP2. BMP2 is a bone morphogenetic protein. And I'll give you a little history lesson about it. In about 1965, this man named Marshall Urist discovered that if he ground up a bunch of bone and stuck it in a muscle, he would get new bone in that muscle. And so it wasn't until about 1971 that he realized that it was proteins in the ground up bone that was responsible for the bone growth. And it wasn't until the late 80s that they identified what these proteins were. And these were BMP1, BMP2, and BMP3 were discovered. And since then, over 20 BMPs have been found. BMP2 is FDA approved for bone formation in clinical applications. And BMP2, bone morphogenetic protein, type 2, really induces bone formations. One of the advantages of it is that there are a lot of growth factors that induce bone formation, but they tend to have a global effect on the bone formation process. As you saw, there was osteoclasts and osteoblasts involved, whereas BMP2 pretty much only affects the osteoblasts. It's already FDA approved, so you don't have to go through any hurdles to use it. The disadvantages are that because it's delivered in a liquid form, it's difficult to localize. It diffuses rapidly, gets cleared rapidly, which requires megadoses for it to be effective. It's not FDA approved in applications like spinal fusion without a collagen sponge and a titanium cage around the sponge. Carriers like collagen can delay the clearance of the BMP2, but they also might release subtherapeutic doses because they tend to bind the BMP2. And the BMP2 has a relatively short half-life, so it may be that once it's released, it's not active anymore. So this is kind of a cartoon of what's going on in vivo if you inject BMP2. It will initiate the differentiation of mesenchymal stem cells into pre-osteoblasts, then into osteoblasts, where they will lay down bone. Now, as I've tried to represent, this bone is very sparse because you get a limited bone response when you inject this way. You need very high doses to get a therapeutic effect. These high peak doses are transient, where a steady constant dose would be optimal. And because BMP2 has a limited shelf life in the body at 37 degrees Celsius, it's not necessarily bioactive for as long. So having a system that was a reservoir delivering BMP2 is not as optimal as a system that would generate the BMP2 constantly. So for this reason, we chose to look at ex vivo gene therapy. And we cloned our BMP2 gene in a plasmid vector. We then packaged it in a plasmid, sorry, not a plasmid vector. We packaged it in an adenoviral vector. We took our adenovirus and incubated it with cells and then injected this into the muscle. Now, these cells continued to release BMP2, causing the chemotaxis and proliferation of mesenchymal stem cells, which differentiated into chondrocytes and pre-osteoblasts and osteoblasts in bone. And we also saw cartilage because this is endochondral bone formation, which is our model. So in our preliminary results, they were very promising. We saw really high transduction with our BMP2 vector, rapid bone formation. Bone was formed within a week. And we saw bone forming in both immune deficient and immune competent mice. Our BMP doses were much lower than if you were to just inject them with a syringe. And the advantages of this system is that it's replication defective, so cells that are implanted into the body can't reproduce the virus, so you can't get it systemically. And we can transduce very many different cell types, which opens the door for clinical applications. And one such clinical application would be spinal fusion. Now, this is an example of a typical procedure involved in spinal fusion. Now, remember that spinal fusion is 2 operations. So first, they'll go and they'll harvest the graft from your hip, and then they'll open up your back, and they'll drill holes where they're going to put the instrumentation. Then they'll place the rods with screws screwed into the holes that they drilled. They'll take this burr and decorticate the bone, which means that they're scraping off the cortex of your bone. Then they'll take the graft that they took from your hip and apply it there. Now, all of this is in an effort to get your bone to fuse together to take over the function of these metal rods. And this would be the final result. Now, you go through all of this, and it's still only 50 to 70% effective. The healing times range from 6 months to 2 years, and sometimes it doesn't work, and you have this result, and then they have to try again. So we decided to try our spine fusion model, our gene therapy model of spine fusion. So we incubated our virus with the cells, with fibroblasts were our choice of cells. We took these genetically modified cells, put them in a syringe, and injected them in the paraspinous muscles of mice. And we saw robust bone formation. In this first image, this is, again, where we injected. This is a normal mouse. This is a mouse that has been injected here. And you can see that it's actually induced scoliosis in the mouse spine. And this is because mice don't ever really stop growing. So what happens is that one side, we only injected in the right side became fixed, and the mouse left side of the spine continued to grow, causing scoliosis. And this is just the micro-CT, and the soft tissue is bleached away here so that we could confirm our radiologic results. And so in order to determine whether these spinal fusions were clinically relevant, I harvested the region surrounding the fusion. I encased it in alginate and then put it in our in-house FLEXION-EXTENSION clamp, putting it in, sorry about that, putting it in both 110 degrees extension and 110 degrees flexion, taking radiological images, taking x-rays of it, and measuring the intervertebral rotation. Now, these images were analyzed with Kimax QMA, which is a software that's FDA approved for use in humans when they're clinically evaluating spinal fusion. And the pink vertebrae are fused, whereas the blue are not. And if I do this, you can see the overlay is changed in flexion and extension, but only in those areas. So if we just look at these, you can see better what the software is measuring. It's measuring the angle formed between these two. And so if the angle was less than a certain amount, it was considered fused. And as you can see, our control and our BMP2 injected animals were statistically significantly different from each other. We got 90% fusion by 2 weeks. Now, if you remember, the clinical fusion is 50 to 70% in 6 months. So here we've outperformed that. So we decided to test our model in a critical size defect model. A critical size defect is defined as a gap in the bone that's too large to repair over the normal life of the individual, and it requires intervention, or otherwise you'll have that gap for the rest of your life. In the long bone, 1.5 to 2 times the bone diameter is considered a critical size defect. There's no real standard with flat bone, so we chose to not look at flat bone. So this is a clinical repair of a critical size defect. This woman was in a very bad auto accident, and the x-ray is kind of fuzzy because it's going through a cast. Surgeons repaired it using plates, and they gave her autologous bone grafts, and they gave her 4 to 6 months to recover from this. So we chose to do a rat fibula critical size defect model. In rats, the models that have been performed are calvaria, which is just the skull, mandible, which is the jaw, the nasal bone, fibula, tibia, femur, and radius. Now, we wanted to do a weight-bearing long bone because they're harder to repair, and we reasoned that if we could repair one of these, we could be sure that we could repair an easier case. The fibula has a marrow cavity. If we could reestablish that as well, then our system was all the better. And because in rodents the fibula and the tibia are joined, fixation is unnecessary. And since we didn't want to do fixation, this only left the fibula for our model. So in our gene therapy repair of the critical size defect, we again incubated our virus with our fibroblasts, collected these cells into a syringe. We created a 2 millimeter defect in the fibula, and then we injected cells into the defect. And these were our results. So the control animal, the critical size defect, remember, won't heal. In the animal receiving the cells, we got bone formation at 2 weeks and more robust response at 4 weeks. And by 6 weeks, the osteoclasts have started to remodel the bone into the proper shape. Now, although this model is working very well, it's got some limitations. It triggers an immune response. Transduced cells will still have virus particles on them, and these can be immunogenic. Non-autologous cells can be immunogenic. And so when we would try these in wild-type animals, it did not work. With the animals that we used, they were either immune deficient or they were so inbred that any cells injected into them were seen as autologous. So in an effort, as you can see here, these were injected with our cells and no bone was formed. In an effort to rescue this, we decided to try microencapsulation. And the benefits of microencapsulation is that cells are localized. They're protected from clearance, from immune attack, but the micrometer size is small enough that you're not going to get cells dying from lack of nutrients because they can exchange waste and nutrients, and they're small enough so that there won't be a core of dead cells. BMP2 can still come out, and these microspheres are small enough that they can be injected and they don't need any surgery to be placed. So the hydrogel that I chose was polyethylene glycol diacrylate, or PEG-DA. And PEG-DA hydrogels are widely used in tissue engineering. They're cross-linked polymer networks that are highly tunable. They've got these physical properties that you can change, that you can tune to match the extracellular matrix of most tissues. You can change their stiffness, you can change their permeability so that they'll let nutrients and waste in and out and keep immune cells out. They're very bioinert. They're extremely hydrophilic. They resist protein absorption and therefore have low cell adhesion. In cases, they've been shown to provide immunoprotection to donor pancreatic islet cells and xenogenic cells when, after they were implanted, they recovered normal function, and so they recovered diabetic animals back into the normal case. So PEG-DA can be used to encapsulate cells. And so if you start off with a liquid prepolymer solution and mix it with your cell suspension, you can polymerize it in many different shapes. Now, the way I created the PEG-DA was I started with polyethylene glycol, and I reacted the hydroxylated end groups from the polyethylene glycol with acrylyl chloride. And this formed polyethylene glycol diacrylate, making both of these ends functionalized. And these acrylate groups will cluster in an aqueous solution because they're very hydrophobic. And in this clustered conformation, they're susceptible to free radical polymerization. And once they're polymerized, you have a hydrogel. And this is how I microencapsulated the cells. I started off with a hydrogel precursor solution in a cell suspension, and I mixed these together. I vortexed in mineral oil and photopolymerized them under white light. And these microspheres, once they were in the mineral oil, I collected them with by centrifuge. And here, our aqueous PEG phase is made of the PEG-DA a hydrophilic photoinitiator, in this case eosin Y and triethanolamine cells, and buffer. The mineral oil phase contains the hydrophobic photoinitiator, which is acetophenone and NVP. And so the goal here, the rationale, is that now these microspheres will protect the cells and they'll form bone even in the wild type case and continue to release the BMP2. But in order to be sure of this, we had to examine the cell viability. And so I ran live dead stains on microencapsulated transduced cells that had been encapsulated after a week. And in the green is the live, because the esterases in live cells will convert calcium AM, which is not fluorescent, into calcium, which is, and it fluoresces green. Ethidium homodimer fluoresces red and only binds to DNA and will fluoresce red once it's bound to DNA, and it only has access to DNA when the nuclear membrane is permeable, and this happens when the cell is dead. So next I wanted to make sure that the cells were surviving injection, and so I injected animals with these microspheres, waited for 2 hours, and then harvested and looked at the structures for damage and the cells for viability. And less than 1% were seen to be damaged, and it was likely due to the harvesting techniques, which were pretty rough. And in the fluorescent image, you can see almost all of the cells are fluorescing green. So they survived. And next was to monitor the gene expression. And so I transduced cells with a protein called DS-RED. And DS-RED comes from the mouth area of a coral. It's a protein that fluoresces. And when you transduce these cells, they will only fluoresce when they're alive. And so it's a way to monitor how long your expression is going on. So we injected these into the quadriceps of mice. And the top row is cells in microspheres, and the bottom rows are cells alone. And in the microencapsulated case, it was the intensity and duration of fluorescence was much higher than in the non-microencapsulated case. And we analyzed with software, so this signal is real, and it's divided by background. So this graph is a quantification of several images like those. The intensity is normalized to background and divided by the mean intensity of the background. So here we've got, in the yellow, the DS-RED cells in microspheres are much greater, statistically greater than the cells alone. And we also tested an empty viral vector cells that were encapsulated. So now that I've established that the cells will survive injection, encapsulation, and that they'll continue to express, in general, the gene expression, now I wanted to establish that they would express a specific gene expression, in this case BMP2. So,
question?
Yes?
So the goal of the encapsulation was to prevent immunoreaction.
Yes.
And you got sort of a byproduct, which was actually more production of BMP2. How did it come about? Why is the microencapsulated cells actually producing more BMP2 than the regular ones?
Because they're not being cleared away. So the regular ones, if you just inject the cells, even in the animals that form bone, they're cleared away within a week.
What is clearing them away?
The body will, even in immune deficient animals, the body sees them, it's not an immune response, but it's just diffusion away from the site, or your interstitial fluids will draw them away, and so they tend to be flushed out of your system.
And the body does not do the same for the microspheres?
No, it can't, the cells can't attach, and they're large enough that they sit where they are. Like, they can't penetrate through, and so they just stay put.
That was because the original goal was the immunoreaction, but you ended up getting something from
Right,
there.
exactly. And so we were pleased with that, because we think we might be able to use even less cells to get a response. And so here, I wanted to make sure that the BMP2 would express for as long as the DS-RED did, because it's a different protein, it's a different adenovirus. So I ran ELISA assays, and ELISA is an assay that'll tell you how much BMP2 is in the media solution that is released by the cells. And so I compared the media from the monolayer of cells that were transduced to cells that were encapsulated in microspheres, and there was no statistical difference across 9 days. The next was to analyze the bioactivity of the BMP2, because the ELISA will only tell you that BMP2 is there. It won't tell you whether or not BMP2 is working. And I co-cultured these with mesenchymal stem cells, and these cells, remember in that little cartoon when you had the BMP2 release, the stem cells were differentiating into osteoblasts and chondrocytes. So these cells will differentiate. They'll start the differentiation process into osteoblasts. And when they do, they release this substance called alkaline phosphatase. And you can measure this. And so if your BMP2 is there but not active, you will not get any alkaline phosphatase activity. So here, these were cells only without transduction. Their media did nothing. The empty virus media did nothing. The monolayer of BMP2 caused a great increase in alkaline phosphatase activity, as did the media drawn from the microspheres. Okay, so now that we've established the system, I injected these microspheres and cells into the quadriceps of mice. And this is an image 2 weeks after intramuscular injection. And these are corresponding images. The first is the micro-CT on the right, and on the left are the x-rays. And so with the bone microspheres, you can just glance at it and tell that the volume went up. However, the density went down. And so we analyzed this, and the tissue mineral content is kind of a measure of how much bone there is. It's in milligrams, so you can think of it like mass. And these were essentially the same between the two, statistically. The volume was statistically greater in microspheres, and the density was significantly less. However, when you think about it, all of those microspheres, they're not degradable, so they're taking up a large portion of that volume, and they're averaging somewhat into the density calculations, so this is somewhat to be expected. So we next applied this model to the critical size defect model, and you can see that the microspheres were forming bone just as the cells directly injected did. And looking at the histology, it confirms our results. We see this pink is bone here. This pink is bone as well. However, in the microsphere case, you can see where the microspheres are because the bone can't form where the microspheres are. We wanted to see what would happen in a wild-type rat, and we injected direct cells, and we injected microspheres. There was no bone formation again when you inject direct cells. However, there was a ton of bone formation with the microspheres. Now, this was good. However, when you look at this, there's a ton of microspheres as well. So this drove the desire to have degradable microspheres. Now, one problem with this is that you don't want to just have any kind of degradable microspheres. If it degrades too slowly, then you're back where you started and you've got big voids in your bone. If it degrades too quickly, you've got too little bone because it degrades before its job is done. So the goal was to have the degradation timed to bone remodeling processes, to bone formation. So if it degrades with an osteoclast, say, that might link it to the bone formation processes. So what I decided to do was time it with osteoclast degradation so that the recruited osteoblasts will form bone first, and later, once the osteoclasts are coming to remodel it, in addition to degrading the bone, they'll be degrading the hydrogel. Now, the best way to do this is, remember, osteoclasts release hydrochloric acid, and they release cathepsin K. So I decided to incorporate cathepsin K degradable peptide into the backbone of the peg. And I've indicated that by this yellow here. With the degradation time to the osteoclast arrival, I have to make it so that the osteoclast can attach. to this PEG. And if you remember that PEG is not, it resists cell adhesion. So I had to incorporate RGDS, which is a peptide that is found in many integrin ligands, and it permits cells to attach. The rest of the polymerization methods were unchanged, but the idea is that the osteoclast will come along, degrade these little yellow blocks, which are the cathepsin K degradable peptide, and now the hydrogel will fall apart. So in order to make the polymer, I first made the peptide, and I added glycine spacers so that once it's sandwiched inside the PEG, the cathepsin K still has access to it. So those are the 3 Gs in front. I added tryptophan because tryptophan will absorb UV at 280 nanometers, and you can use that to measure its release into media and see if you're actually degrading your peptide. And so I reacted the free amines on my cathepsin K degradable peptide with heterobifunctional PEG. So in this case, I chose acryl oil, PEG, and hydroxysuccinimide, and the free amines on the peptide will form peptide bonds with the active esters of the NHS. And if you include lysine, which I did, lysine has free amines on its side chain, and so that enables this peptide to be sandwiched and have PEG acrylates on either side. Now, in order to synthesize PEG RGDS, I reacted with acryl oil, PEG, and hydroxysuccinimide, and this I reacted only on one side so that I had pendant chains of PEG RGDS, and this gave it more bioavailability to the osteoclasts. So first I just wanted to test and see whether or not my peptide was capable of being degraded by cathepsin K, because if it's not, then the hydrogel won't be. So I suspended my peptide in buffer, and I analyzed under mass spectroscopy. And the predicted molecular weight was about 1144 Daltons. The observed weight was just about 1144, so it was there. When I added cathepsin K, that peak disappeared, and this peak was replaced by predicted degradation products, confirming that our cathepsin K degradable peptide was indeed cathepsin K degradable. Here, I formed hydrogel structures and incubated them in different buffers. The first, at the bottom, is the sodium acetate buffer, which is our negative control. And then proteinase K, which is the positive control, should degrade the peptide as well. And cathepsin K, which is what I want it to degrade in, degraded the peptide. And so the way that I evaluated this is tryptophan was a part of the sequence. When the peptide is cleaved, it is released into the solution. Otherwise, it should be bound in the hydrogel. And so as you can see, the measure of tryptophan release, our hydrogel completely degraded. I couldn't see it anymore in the cuvettes as well. And then next step, okay, it's degradable by cathepsin K, but that doesn't mean it's degradable by osteoclasts. So I formed hydrogel sheets and seeded these with osteoclasts and labeled them. I labeled the osteoclast with dapian phalloidin and the hydrogel with Alexaflor 680. I washed away the cells at 48 hours and imaged the osteoclasts. And this ring of death is pretty much the typical sealing ring that you see in an osteoclast. And osteoclasts will form stereotypic or characteristic pits. And I saw these on the hydrogels afterwards. So I wanted to make sure that this wasn't just something that any cell seated on these hydrogels would do. So I formed hydrogel sheets with mica because mica has an atomically smooth surface, and then seeded osteoblasts and osteoclasts on hydrogel sheets, and then imaged them, and after washing them off first. On the osteoblast surface, you can see it was completely unblemished. On the osteoclast surface, after 24 hours, we see some of the pits that are characteristic of osteoclasts. And this is a larger magnification of one of those pits. Now, this is a movie of the fluorescent labeled osteoclast on a gel and the green gel underneath. If you look, you can see where it becomes dark. That's where there's less fluorescence. That's where the hydrogel has been degraded. And this is a 3D reconstruction. If you wait and watch, you can see in the combination of the two, you can see through the hydrogel and you can see the osteoclast on the other side. When it's separated, you can see that that dark region is actually transparent here. So all of this is suggesting that our osteoclasts are degrading our hydrogel. So we compare against the osteoblasts. And you can see that there are no dark regions underneath the osteoblasts. So the osteoblasts aren't capable of degrading the hydrogel the way the osteoclasts are. So now that we've determined that cathepsin K can degrade the hydrogel, osteoclasts can degrade the hydrogel, I want to see what happens in vivo. And I injected these degradable microspheres in the quadriceps of mice and analyzed after 3 and 6 weeks. And so far, the bone looks more mature than what we've seen before. Not really more I can say about that until I get histology back, because these are pretty recent results, but I can say it looks different. So in conclusion to this work, our system is osteoinductive. We induced the alkaline phosphatase activity in the stem cells, which showed that they were being induced to differentiate. It's osteogenic. You all saw that it created bone formation, and it's bioresorbable. We'll have to wait on histology to determine whether it's osteoconductive, but it's a possibility. So the potential clinical impact of that is that perhaps one day we can go from this highly invasive procedure that only works half of the time, takes 6 months to recover from, to maybe a series of injections for spinal fusion. Another potential clinical impact is this. This is Roger Ebert, who's a film critic. He lost his jaw due to complications from surgery. And they went in and they tried several different autografts. They took bone from his hip, they took bone from his scapula, his shoulder blade, and all of them failed. And they can't take bone anymore he broke his hip because it was so weakened and he can't sit upright. And so this is what he's left with. And perhaps one day we can restore him to what he looked like before through this method. Now, for future directions, one of the things that caught my interest was I saw this is a group that got stem cells to mineralize in the presence of BMP2. And they modeled it, starting with random initial conditions like these. Now, you can take what are called these diffusion reaction equations, and you can model biological activator-inhibitor or activator-substrate systems. Now, these particular equations are Gray-Scott equations, and this would be your activator, in this case BMP2, and this would be your inhibitor. And so I thought to myself, what if I try to do that on the modeling of my microsphere case? And let's say I just looked at that area, and I took out everything but the bone formation. I know my initial and boundary conditions. My histology will enable me to compare against my model in the 2 dimensions, and micro-CT will enable me to compare against the model in 3 dimensions. And so hopefully what I can do is I can model this bone formation and describe what's going on, and then use that to design maybe the size of the microspheres or how many are in a place, because we can always up the solution or alter those conditions. And so then maybe if I model this, I can better design our gene therapy system. Now, one of the other things that has caught my interest, and this is part of the work that I'm doing with the NSBRI, is in 1931, these archaeologists found this jaw with 3 teeth in it that weren't teeth. These 3. They were pieces of nacre, and they assumed that the nacre, which is mother of pearl, the inside shiny, pretty part of a shell, they assumed that these had been put in posthumously. And it wasn't until 40 years later, in about 1972, that a dentist happened to be in the museum where this jaw was, and he asked them if he could x-ray it. And he noticed that there was actual osseous integration into the bone of these nacre teeth. So the ancient Maya had the very first endosseous implant recorded, and they pretty much have better than what we get today. So that caught my interest as well, as you can imagine. Nacre forms mineralization. It's a biomineral through completely acellular processes, whereas bone does not. Yet some of the patterns are so strikingly similar. If you magnify nacre even more, its pattern has been likened to what's called a Voronoi simulation. And so the Voronoi pattern here, it starts off with these tiny nucleation points, which are allowed to grow. And when they intersect each other, that's where you get the boundary lines. So there were a group of researchers who were also intrigued by the Mayan teeth, and they decided to see what osteoblasts alone would do. So they took a fragment of nacre and a fragment of bone and put them on a layer of osteoblast. Now, near the bone fragment, bone grew towards the nacre. Now, near the nacre fragment, nacre grew towards the bone. And so what they've been trying to do is they've been trying to tease out what in the nacre is causing this result. I became interested in this, and I wanted to tease out what in the osteoblast has changed to cause this result. So I repeated their experiments, and I got really interesting results. So this is where my bone is, and that schematic I showed you, and this is where my nacre is. And if you don't really know what bone or mineralization on a layer of osteoblasts look like, it looks like little random, let's say you were eating an Oreo cookie and you left crumbs. That's what it looks like. Here, it looks very different from this on a global scale. It's looking more like it's following that pattern. And on a small scale, it looks like it's following that pattern as well, which is not what they do. Here you can see at 2 different time points, this mineralization is increasing in size. And here, I don't know how well you can see that, but this top square or rectangle is nacre, and the bottom is rectangular bone, which should not happen. This is the shape that the bone usually takes. So I decided I'm going to analyze this, and I looked at the morphology under SEM, and the new mineral resembled bone more than it resembled the other. And I analyzed with energy dispersive analysis of X-rays, and you can look at the basic elements that are in there with this method. And nacre and the outside layer of the shell, which is the prismatic layer, are made of calcium carbonate and do not have phosphorus. However, bone is made of hydroxyapatite, where there is phosphorus. And the new mineral, although it's not exactly like bone, it looks different than the nacre. The nacre and the prismatic layer of the shell do not have the phosphorus. One of the other differences is there's a peak in there at 868.36. And that peak disappears only in cultures where this altered mineralization happened. So I'm trying to tease these things out. And the reason for this is nacre's 3,000 times stronger than the geologic version. Nacre is an isomorph of a polymorph of calcium carbonate called aragonite. And geologic aragonite is nowhere near as strong as nacre. And the reason for this strength is its structure. It has what's called a brick and mortar structure, where it's got interdigitating layers. And if I can somehow direct bone to form in the pattern that nacre is forming, perhaps I could use bone to be its own fixation, so you wouldn't need cements and you wouldn't need pins and rods and things like that. And so one of the things that I'm interested in doing in the near future, as opposed to the far future, is pegylating one of these proteins and patterning some hydrogels with them and seeing if I can direct the biomineralization to occur in the patterns that I set. Because ultimately what I want to do is I want to use clues from nature to help to design the technologies of the future. And with that, I thank you for your attention. Do you have any questions?
Perhaps I missed something at the end there. Are you saying that the presence of nacre causes osteoblasts to lay down bone in a different pattern that more resembles the motor structure of the nacre? Than
Yes.
So it's as if you are seeding a material like a crystal based on the underlying structure?
Something like that. It's not something that happens every single time. And so that's why I'm really excited that I found that protein, because I want to figure out what is it that's causing it. And it could be location of the shell, because there's a growth front on the shell. It could be, there's so many different things that to tease out, but it has happened enough times. And I do several cases. I do cases where it's just the media, cases where it's bone, and nacre. It only happens if nacre is present.
So if it were being induced by the underlying crystalline structure of the material, you'd expect that the effect would dissipate with distance.
Right, it happens closer to the nacre than anywhere else.
How far away from the nacre can you get nacre-like structures of bone?
I haven't seen it. Let me see. Let me go back. I haven't seen it much further than 1 millimeter away. So if this is that whole distance, I haven't seen it much further than like that radius around the nacre.
So to make this work, you would seed the material with the nacre to sort of...
I want to find the protein in particular. The first step is to just, I can mix the nacre with the hydrogels. I can mix like a bunch of proteins. But the next step is there's a specific protein in nacre that has been linked to biomineralization, and so I'm going to look at that protein first. That's a starting point. But I'm also doing micro arrays of the RNA in the osteoblasts that caused that odd mineralization versus osteoblasts that did not, and proteomics as well.
I also have a question in terms of the translation between the animal model and clinical problem.
Mm-hmm.
So what, and I can imagine two different hypotheses. One, that the problem in the human is insufficient induction of the osteoblastic activity, which your encapsulated and releasing mechanism might overcome.
Mm-hmm.
But the other mechanism, of course, is insufficient mechanical fixation during a process that's already set off well biologically, but it's a large animal and it's rather nasty bone arms and spine.
Right.
Any sense of translatability with the animal?
Well, the thing about people is that you can tell them not to move, and you can't really do that with the animals. You can cast something, and instead of then going in with the plate, because that won't, the critical size defect, even if you cast it, that won't heal. You wouldn't necessarily, if this worked, you wouldn't need to fix it. You would just need to inject and then cast and tell the person, don't move your leg. With spinal fusion, I envision, now I'm not married to this, I'm not sure that I'm correct in any way, but I envision that you could, if this were to work in people, you could brace them so that they would stay still for that. Because if you have too much movement, you're not going to fuse, you're just going to form a non-union. But, again, I'd have to see it in an animal that is upright as opposed to crawling on all fours before I made any conclusions.
So I'm curious about the computational approach you suggested, I think what we call Gray-Scott equations.
Mm-hmm.
These kinds of equations, when you have an activator and an inhibitor, will lead to different shapes,
Yes.
strongly dependent on parameters.
Mm-hmm.
Do you know You tested the equations to see if you found to get different shapes, and if the shapes had any relation to what you get in the bones. I mean, do the bones even obey these equations?
Well,
And one more
okay.
question. What would be that inhibitor? BMP2 is the activation.
Oh, it's I think it's it's MLA or MLGA. I can't remember the name of it. I can find that out for you though. But it was in their model. It was genetically modified stem cells, and the inhibitor was incubated with them. With the patterns, yes, you can alter them, and yes, they look. You can make them look like fingerprints. You can make them look like coral. You can make them look like bone. It really all depends on the parameters you choose, and you can have it as complex as you want, where the parameters are still physical, or you can have it so simplified that they're just non-dimensional parameters. And so it's definitely a challenge, and it's something that has interested me. However, I haven't had a chance to pursue it further than just trying out different parameters and seeing what I get.
Can you control those kinds of parameters experimentally?
I can control the size of the microspheres that I inject, and I can control the concentration that I start out with, because I know that if I have a rough estimate of cells per microsphere, then I'm going to have roughly this much BMP2. And so if I assume that nothing else is correct and manage to model it so that it at least looks like the histology, if then I only change the size of the microspheres and then I get a drastic change, then I think that will tell me something.
So to respond to your question, that work was published by the Venus group in the City National Capital, where they used the mesenchymal stem cell, which we grow as well in our lab. What kind of nodules?
Right, right. Well, do you remember the name of the inhibitor? I don't remember the name of the inhibitor.
I don't remember.
Yeah, I can find out.
We are able to model this morphological patterns. This mathematical model. It's very interesting phenomenon. Some of the
I
processes
think this
occur
is very
while
useful,
resting.
because if you suppose you can prove that you can get, these equations appear in different forms in different fields, and you can see the shapes appearing. So the first thing is to show that in fact you can get those various shapes so that you validate the equations, and then of course in the end you have to use the equations to find the right parameters for the problem at hand.
Right, right.
So that becomes really interesting.
Because it does simple one, that same pattern applies to blood vessel as we age, we become calcified. But instead of using BMP2, we deal with BMP4.
Right.
In your micro-encapsulation model, all the tryptophan are released in 3 hours. So that seems to suggest that if you could just protect the vectors with something like a pitted titanium oxide nanotube for 3 hours, that might be enough to help it a lot in the future.
But it's degraded in the hydrogel is degraded in 100, well, in a solution of cathepsin K in 3 hours, but not by osteoclasts in 3 hours. And so after 6 weeks, like, I still have to see the histology, but after 3 weeks, hydrogel was definitely still there.
That was a great talk. Could you comment on the use of gene therapy? You use adenovirus.
Right.
So I noticed that your X-ray after encapsulation, the morphogenesis seems to be more regulated as opposed to.
I didn't hear you.
When you look at the X-ray at day 6, the contour of your cortex of the bone seems to be more regulated as opposed to direct gene therapy. Since we know that osteosarcoma does occur with this kind of treatment, so do you think encapsulation is a means to prevent complication from gene therapy?
I think it can be. I think that, it's difficult to say if during the encapsulation process and then the washing process, I can't 100% say that there are no particles trapped and embedded in the peg. It is washed, and the surface tension, I imagine, but it's a possibility. Like I can't say for sure. I mean,
You mentioned about timing the duration in osteoclast. So is the duration in osteoclast or introduction of osteoclast critical to control harness release of BMP2?
wait, ask.
Because you have encapsulated
Right.
BMP2 so the osteoclast will be protected from
Well, it's not encapsulated osteoblasts. They're fibroblasts in our case. We haven't used osteoblasts. The osteoblasts are recruited from the host. And the encapsulated cells, their only job is to release BMP2.
But there's got to be a control as to how long BMP2 should be released.
Right.
Instead of having normal bone formation, you will have cancer.
Right, right. Oh, I'm sorry. We've also, I didn't present it here, but we've also formed a TET on and TET off BMP2 vector. And so when you add into the system tetracycline, then you can turn it on or off. And so it can be controlled, and we have controlled it. And that's one of the things we're looking at as well. It seems like as long as you have it for a week at least, then you get bone formation. Now, the next is to tease out exactly how long it needs to be, because I'm certain it's different in different animals. Yes?
Can you talk about the mechanical properties of the bone constructs?
Well, I could tell you about the spinal fusions. As far as the spinal fusions went, the software that we used to put it in flexion and extension was the same that they use to evaluate if a person has a clinical spinal fusion. And so other groups will also do three-point bending or things like that. We were more interested in seeing clinical relevance because you can't really remove a human spine and do three-point bending. With this software, it essentially just showed the rotation, whether or not there was any rotation when you put it in flexion and extension. And in fused spines, the rotation was within the error of the system.
Thanks very much.
Thank you.